Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Ditoro Makwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So in today's lesson, we are going to be looking at our final exam preparation, and we will be focusing on meiosis. So here we are just going to look at the process of meiosis and the phases that are involved in the process of meiosis and how meiosis is important. Okay, so because we are looking at our final exam prep, just remember the structure of the paper that you're going to be writing. This is a paper out of 150 for 2.5 hours, and we have section A as well as section B. In section A, we have 50 marks for our short questions such as multiple choice, terminologies, as well as the matching column. And in section B, we have question 2 and 3, which are going to give us 100 marks. Now, by the end of our lesson today, you should be able to list the two types of gametes, right? Those are our sex cells. You should also be able to describe the karyotype of humans, meaning that you should be able to differentiate between the karyotype of a male and a female human. You should also be able to describe the number of chromosomes in somatic and sex cells. So the difference between the number of chromosomes in these two cells. Next, you should be able to differentiate between haploid and diploid chromosomes, as well as to describe the process of crossing over. Remember, crossing over is very important during meiosis, so we are going to describe that. All right, so let's have a look at our pre-knowledge. So here for your pre-knowledge, you have to have some knowledge of mitosis. This means that you should be able to differentiate between mitosis and meiosis. Okay, now let's have a look at a baseline assessment. So for our baseline assessment, we are just going to be labeling the structure of the chromosome that we are looking at here. So we have two chromosomes, as we can see. At A, we have the chromosome itself. So A here would be the chromosome. And then at B, we have the centromere. Remember, the centromere is the part that holds the two chromatids together. Next, we have C, which are the chromatids. Remember, one chromosome is made up of two chromatids. So these are the chromatids. At D, we have homologous chromosomes. Remember, when we talk about homologous chromosomes, we are talking about two chromosomes that whereby one is of the maternal origin and the other is of the paternal origin. As we can see, we have shown that they are of different origins with the color. So you should be able to label this because questions based on meiosis may be confusing if you don't understand the difference. If the question is asking about the number of chromosomes, if it is asking the function of part B, for example, when you are referring to chromatids and when you are referring to chromosomes, there should be that understanding of how chromatids differ from chromosomes. There should also be an understanding of how homologous chromosomes differ from a normal chromosome. So there we have our labels for A to D, right? And then for our terminologies are also very important, right? So the first one that we have is haploid, which is having just one set of chromosomes in the nucleus in each cell. And then we have diploid, which is having a double set or two sets of chromosomes in the nucleus of each cell. So remember, haploid is half of diploid. If, for example, in humans, our diploid number of chromosomes is 46, in the haploid number, we are going to be looking at 23, which is half of 26. And then we have autosomes. You should be able to differentiate autosomes from gonosomes. Autosomes are all the chromosomes except the sex chromosomes in the cell, right? And then the gonosomes would be the chromosomes, the sex chromosomes, which is usually pair number 23 in humans, which means that because autosomes are all the chromosomes except the sex chromosomes are gonosomes, meaning that here our autosomes in humans would be 44, right? Which is excluding pair number 23. Okay, and then we also have karyotype. This is the number and structure of chromosomes in the nucleus of a cell. With the karyotype, you may be asked to define this terminology in the examination, so make sure that you know it. Okay, and then we have gametes. Here we're talking about the term for haploid sex cells. Sex cells and gametes are the same thing. Now, when we are talking about gametes being sex cells, you should then be able to understand that sex cells and sex chromosomes are not the same thing, right? Cells are a cell, which may be your sperm cell and your ovum. Those are sex cells. However, within any cell, we have sex chromosomes, right? Which, as I said, those would be usually pair number 23 in humans, whereas the cell is the whole cell, which has its own number of chromosomes. Okay, somatic cells, the normal diploid body cells. Somatic cells are the body cells, and then the sex cells are the ovum and the sperm cell. Crossing over, a process involving the exchange of genetic material between members of a homologous pair. Homologous pair refers to a pair of identical chromosomes, one of paternal origin and one of maternal origin. And then we have variation. We're going to talk about variation when we talk about crossing over, because crossing over brings about genetic variation. So this is the variety of appearance shown by organisms of the same species. Okay, so now, as we were talking about somatic cells and gametes or sex cells, the somatic cells and the sex cells differ when it comes to the number of chromosomes that they have, right? Remember, the body cells are somatic cells, sex cells are gametes, right? Now, the number of chromosomes in the somatic cells are diploid, and the number of cells in the gametes are haploid, meaning that in the somatic cells, we see that there is 46 chromosomes, whereas in the gametes or in the sex cells, then we see that there is only 23 chromosomes, which is half of the number that we have in the somatic cells. Hence, we say that gametes have half the number of chromosomes. They are haploid, whereas somatic cells are diploid. Okay, so the gametes, as I said, we are referring to the ovum and the sperm cell. So make sure that you remember that the ovum and the sperm cell are our gametes or our sex cells, right? Then why do we experience meiosis? Why do the cells in our body experience or go through meiosis? Why is meiosis so important? We're going to be looking at three importances of meiosis. The first one being that it is a mechanism that introduces genetic variation. How does it introduce genetic variation? Through two things. During crossing over of the chromatids of the homologous pair of chromosomes, we're going to look at the fact that they're going to exchange genetic material, meaning that the original chromosome are going to be changed after crossing over. And then the random arrangement of chromosomes at the equator. Also going to look at that when we look at metaphase. This takes place during metaphase. Remember, variation is important because it makes sure that the organisms of the same species are able to survive when the environmental conditions are changing, right? And then to prevent things such as extinction of the species. And then secondly, meiosis prevents the doubling effect of fertilization. Remember, when fertilization happens, we have N plus N, which gives us 2N. Now, if this 2N does not undergo meiosis to become N, then, because remember, meiosis halves the number of chromosomes, then it means that we will have 2N plus 2N, which will then give us 4N. This is the doubling effect. So because of meiosis, we have 2N, which will then undergo meiosis and ends up as N. Then this N from the other individual will combine with the N that has undergone meiosis, and this N also undergone meiosis, giving us 2N again. So essentially, meiosis keeps the chromosome numbers the same from one generation to the next. It makes sure that the chromosome number remains the same. Lastly, meiosis results in the formation of haploid gametes. Remember, haploid simply means cells with of the chromosome numbers. So it makes sure that the gametes or the sex cells are haploid, which we are referring to the sperm cells and the ovum. Okay, now looking at the phases, remember here we have interphase, which is the phase that takes place before meiosis in preparation for meiosis, right? So as you are looking at these phases, just remember that you should be able to identify it by looking at it, and you should also be able to describe what happens during that phase. Now, the phase is interphase. The processes that happen during the phases should not be confused as the phases. For example, during prophase, we experience crossing over. That is a process that happens during the phase of prophase. Okay, so the main process that takes place during interphase would be DNA replication, whereby the chromosome numbers are going to be doubled. Okay, and then in prophase 1, remember we have meiosis 1 and meiosis 2. In prophase 1, the main event that takes place here would be crossing over, right? The main event taking place here is crossing over, as we can see here. And we also have the nucleolus and the nuclear membrane disappearing. Those are some of the things you could be on the lookout for when you're trying to identify the phase. And then another thing that you should not forget is the fact that we have these, which are the centrosomes, when before they separate into centriole. So you should be able to differentiate that as well, right? So this is the centrosome, right there. And then when the two separate to the opposite poles, when we have them here, they are now referred to as the centriole. So be able to differentiate between a centriole and a centrosome. Now, the details of crossing over. Remember, with crossing over, we are going to look at the fact that these chromosomes are going to lie next to each other, and then the chromatids, the non-sister chromatids of the homologous chromosomes are going to overlap, right? When they overlap, they are going to exchange genetic material, and the resulting chromosomes are going to be different because they will be containing segments of the other chromosome. So this is how you should then be able to describe it if you are asked to describe it in the exam. It occurs during prophase 1. Homologous chromosomes or chromatids overlap at a point called chiasma. Remember, the point called chiasma is right here, which the plural word would be chiasmata. And then the genetic material is exchanged, resulting in a new combination of genetic material. Metaphase 1, the main event that takes place here is the random arrangement of chromosomes. So these chromosomes, as you can see, the paternal one or the maternal one is on one side, and here it's on the other side, meaning that the chromosomes are arranged randomly. Not that all the green ones should be on one side and all the blue ones are on one side. There is that mixing of genetic material here as well, or the mixing of the chromosomes, so that they are not on the same side and ends up being pulled to the same poles when the chromosomes separate. Again, how do I identify that this is metaphase 1? The chromosomes align at the equator in homologous pairs, whereas in metaphase 2, you will see they're going to align individually or singly. Again, we have the spindle fibers. Remember, the spindle fibers are the ones that hold the chromosomes at the equator. Okay, so how you identify metaphase 1 is that the chromosomes are aligned at the equator in homologous pairs. Anaphase, the spindle fibers will contract, the chromosomes will be moved to the opposite poles. They're going to be pulled to the opposite poles, right? And then we can see this cell will have a mixture of genetic material, and this cell will also have a mixture of genetic material. So crossing over, as we can see the different colors, together with a random arrangement of chromosomes would have then led to the cells containing different genetic material. Okay, now we have two cells that have resulted from this when we look at telophase, right? We have the two cells forming on both poles, and then we can see that during telophase, the cytoplasm is going to divide so that the two cells are going to result from that, right? Now we have two groups of chromosomes on each pole, right? And then it results to the formation of nuclei on both ends, meaning that we can now start to see that there is a cell, two cells that are found on each end, right? And then we can see here, there we have our full cell, two full cells that have half the chromosome numbers as initially. So if we look at this diagram here, we will see that the cell started off with 1, 2, 3, 4 chromosomes, but now each cell only has 2 chromosomes. So the halving of chromosomes happens during meiosis I, right? Each cell now has half the chromosome numbers as the original cell. The original cell had diploid chromosomes. Now the cells have haploid chromosomes. Okay, let's have a look at an activity. Activity 1. It says the diagram shows a process during meiosis. The letters represent the alleles of genes in the chromosomes. Okay, as we can see here, we have the numbers which are, or the letters which are representing those alleles. We have 1.4.1 to 1.4.4. 1.4.1, name the process shown in the diagram. We can see that there is crossing over, so that is what is happening here. And then the phase of meiosis represented above, that would be prophase 1. Okay, and then 1.4.2, identify point D, that is chiasma. Okay, and then we have B, identify part B. Part B is a chromosome. Remember, if it was all of them, it will be a homologous chromosome. But if it's just one chromosome, then we just refer to it as a chromosome. 1.4.3, write down the letter and name. Be sure to make sure that you write both here in order to get your 2 marks. The letter and name of the part that join together two chromatids. That joins the 2 chromatids together. Remember, that is part C, and we said that it is a centromere. So it means you get a letter and a mark for saying C, and you get a mark for saying centromere. Always make sure that you check the mark allocation and you check whether they want both the letter and the name, just the name or just the letter. 1.4.4, write down the genotypes of the resulting chromatid A after the process named in 1.4.1a. So chromatid A, here it is. So chromatid A initially was R, T, N, D, the black part. Now it is going to be swapping with the white part, meaning that now we are going to have R, T, N, D. So this would be our answer for the new genetic material of chromatid A. Okay, so we have R, capital T, small n, capital D. So this can be in any order as long as all those letters are represented, they are visible. Okay, moving on to meiosis II. We have prophase II. Now, we do not have interphase before prophase in this case. Interphase takes place before the whole process of meiosis takes place. So during prophase II, it differs with prophase I because in prophase I we have a crossing over, whereas in prophase II we do not have the crossing over. So here we're just going to see that the chromosomes are going to become distinct. We're going to see that the nuclear membrane starts to disappear, as well as the nucleolus, right? So this nucleolus will also start to disappear. Now we can see the chromosomes as they are going to be looking distinct. Now that centrosome that I spoke about earlier, it is now going to form the 2 centrioles, meaning that we can also start to see the spindle fibers reappearing. Okay. Metaphase 2, same as metaphase 1. Chromosomes are going to align randomly at the equator. The only difference here is that these chromosomes will be lining up at the equator singly. As you can see, initially in metaphase 1, we looked at the chromosomes aligning at the equator in homologous pairs. Now they are going to be aligning singly and randomly, meaning that it doesn't mean that this chromosome has to have this segment that is mixed on the left or on the right, or all the chromosomes having the mixed segment on one side. It is just going to be a mixture, and that means that when the the two new cells are going to be formed. It is going to be random as well, meaning that it leads to the mixture of genetic material here. Anaphase, again, the chromosomes are going to be pulled to the opposite poles. In this case, we are pulling chromatids, a single chromatid, as opposed to a chromosome. So the chromatid is going to be pulled here, meaning that this full chromosome is going to be split into two and it is going to be separated like that. Those chromatids will then move to the opposite poles, meaning that we are now going to see that the chromosomes are going to form at the end of each pole, and we are going to experience the formation of new cells. Now, these are still referred to as the number of chromosomes. We refer to them as unreplicated chromosomes, meaning that we are allowed to say it's chromosomes, meaning that when you are asked how many chromosomes, you don't look at the fact that it's just chromatids. They are still chromosomes. So you will count those chromatids and you as chromosomes, and that's the number you will give. Don't say, okay, this is not a chromosome. As they are looking like chromatids, they are referred to as chromosomes. Okay. Then telophase 2, remember with telophase, we're going to be looking at the cytoplasm dividing and allowing the two cells to form. We started off with one cell. At the end of meiosis I, we had two cells. Those two cells experienced meiosis II, and now they are splitting. Each cell is splitting into 4 cells. So we know this. We know that at the end of meiosis, we expect to see 4 cells, and these 4 cells are going to be genetically different from each other. Now let's look at what makes them genetically different. Here we have one chromatid that has a green one that has a blue segment, and then this one, it has blue and green. These two, they are plain, so these two cells are different. And then this cell is a bit similar to this one. However, this one is different from this one as well, because this one, the bottom part is blue. The original one was blue, and then there was a mixture of the blue and the green. This one is mostly blue with a bit of green. This one is mostly green with a bit of blue. So this shows us that these chromosomes are different from each other, leading to our cells being genetically different from each other. Now, if these 4 are the 4 sperm cells that are produced, it tells us that these 4 sperm cells are going to be different from each other, which is what is going to lead to genetic variation of the organisms of that species. Okay, so the 2 cells have undergone meiosis II, and they have resulted in 4 cells. Main thing here is that those 4 cells are genetically different from each other with half the chromosome numbers. Okay, now let's have a look at our second activity. It says here, the diagram below represents a cell during a certain phase of meiosis. By looking at the cell, you should be able to see which phase this is. As we can see, we have 4 chromosomes that are full chromosomes, and they are aligned at the equator in homologous pairs. It tells us that this is metaphase 1. Also, when you are asked to name a phase, be specific. Is it metaphase 1? Is it metaphase 2? If you do not specify, you will lose those marks. So 331, identify the phase of metaphase represented in the diagram above. This is metaphase 1. 332, give one visible reason for your answer in 331. As I said, the chromosomes are aligned at the equator in homologous pairs. So that is what tells us that this is metaphase 1. Okay, 333, describe the process that caused the inheritance of a combination of both parental characteristics in chromatids A and D. The process that led to this combination of characteristics in the chromatids, that is crossing over. The process is crossing over. But remember, the question says describe. So you don't just describe, you don't just name, you must name and describe. Because they didn't tell you that this is crossing over, you will get a mark for mentioning that it's crossing over and then describing what happens. As we looked at the description earlier, you're going to describe the fact that the chromatids are going to, the chromosomes are going to lie next to each other, the chromatids are going to overlap, genetic material is going to be exchanged, right? So during prophase 1, chromatids of homologous chromosomes overlap chiasma. Whenever you talk about crossing over, make sure that you mention chiasma, right? And exchange genetic information between them. This process is called crossing over. So if any process is not mentioned, the name of the process is not mentioned, make sure that you mention the name to score a mark for that. If it has been mentioned, there is no need to give the name, but make sure that you can always just mention it just to be safe. 334, state 2 reasons why these chromosomes are homologous. As we can see, the chromosomes the same length, they are of the same shape and size, and their centromere are at the same position. So it tells us that they are the same, right? Or they are homologous. And then we also have the alleles for the same gene at the same position, as you can see over there. So we can say that these two are homologous chromosomes. Now, just to summarize what we discussed, in meiosis 1, we have the different phases there: prophase 1, prophase 2, anaphase, and telophase 1. Telophase and cytokinesis, which is the division of the cytoplasm, right? Whereby we end up with 2 haploid cells. The chromosomes number have gone from 46 in humans, or 2n, to being n, which is 23, right? And then in meiosis 2, with prophase 2, metaphase 2, anaphase 2, and telophase 2. Again, make sure that you master the main events that take place within each phase, right? Here, the sister chromatids are going to separate, not the chromosomes, not the homologous chromosomes separating. Now it's going to be the sister chromatids. Okay, now in our next lesson, we will be looking at the consequences of abnormal meiosis. So we've looked at how meiosis would normally take place. Now we will look at what happens if meiosis fails to take place normally, the consequences of abnormal meiosis and what it results in, which is basically Down syndrome, right? For your homework, we have another diagram on meiosis. Make sure you go through it so that you can be able to make some corrections during the lesson. Okay, we have come to the end of our lesson for today. Thank you so much for watching. I will see you next time. Goodbye.